In order to understand the catalytic properties of enzymes, we have to actually understand what takes place inside the active sites of enzymes. And to gain a better understanding as to what takes place inside the active sites, we're going to study a specific example of a protease that is found inside our body, namely chymotrypsin. So chymotrypsin is actually a serine protease that is found inside the small intestine, which basically hydrolyzes or catalyzes the hydrolysis, the cleavage of specific peptide bonds. It cleaves on the carboxyl side of relatively large and bulky hydrophobic side chain groups, and this includes methionine, phenylalanine. It also includes tryptophan and tyrosine. So if we take a look at the following hypothetical polypeptide chain that consists of 7 amino acids, we have glycine, methionine, phenylalanine, tyrosine, alanine, tryptophan, and glycine. And this is the corresponding three-dimensional structure. Now, let's take any one of these amino acids. So let's suppose this amino acid here, and this happens to be the tyrosine because this is the side chain group of tyrosine. Now, this C carbon here is the central carbon atom. This side is the carboxyl side, so the right side is the carboxyl side, and this is that carboxyl peptide bond. While the other side is the amino side, and this is that amino peptide bond. And what chymotrypsin does is it only cleaves methionine, phenylalanine, tyrosine, and tryptophan on the carboxyl side, on the right side of that amino acid. So if we examine, let's say, methionine, so this is our side chain group of methionine, this is the green bond that will be cleaved by the chymotrypsin. Likewise, if we move on to phenylalanine, this is the phenylalanine. So on the right side, the carboxyl side, is where chymotrypsin will cleave that bond. Again, it's shown with green. Let's move on to tyrosine. This is tyrosine. This is the bond that will be cleaved here. And finally, tryptophan. So chymotrypsin cleaves on this side of that bond. Now, let's take a look at alanine. Well, because alanine is not a bulky hydrophobic or aromatic amino acid, what that basically means is this bond will not be cleaved by chymotrypsin. Likewise, this bond will also not be cleaved because glycine is not a bulky hydrophobic or aromatic amino acid. So, chymotrypsin is a serine protease that catalyzes the breaking, the cleavage of peptide bonds on the carboxyl side of bulky, so large, hydrophobic or aromatic amino acids. Now, chymotrypsin is a serine protease, and chymotrypsin is an example of an enzyme that utilizes the covalent catalysis mechanism. And what that means is, inside the active site of chymotrypsin, we have some type of nucleophilic residue that acts as the catalyst in forming a covalent bond, a temporary covalent bond, between the active site of the enzyme and that peptide substrate molecule. Now, we say that the covalent bond is temporary because at the end of the reaction, that bond is broken. And that's important because we have to reform that unmodified enzyme. We have to regenerate that enzyme at the end of any enzyme catalyzed reaction. Remember, enzymes are never changed or depleted at the end of the reaction. They always have to be regenerated. And that's why this bond is only a temporary covalent bond. Now, we said earlier that chymotrypsin is an example of a serine protease. And what that means is, inside the active site of that chymotrypsin, it's the serine molecule, the serine amino acid, that plays the role of the powerful nucleophile that catalyzes the cleavage of that peptide bond. But how do we know that the active site contains this serine atom, serine molecule that plays the central role in catalysis? Well, basically, the way that scientists discovered this is they probed the active site with a specific type of irreversible inhibitor. So they used the specific group inhibitor known as diisopropyl phosphofluoridate, or DIPF, and this is the same irreversible inhibitor that we spoke about in our lecture on irreversible inhibitors. So If we take DIPF and we mix it with an enzyme, and inside the active site we have a serine molecule, and if the reactivity of that serine molecule is quite high, then that DIPF will basically form a covalent bond with that oxygen of the serine, displacing the H, and that will kick off this fluoride atom, and we form a covalent bond between the oxygen and the phosphorus atom. And once we create this covalent modification, the irreversible inhibitor basically blocks the activity of that enzyme. And so we know that because the activity of the enzyme is inhibited, that means that the DIPF must be bound to a serine found inside the active site. Now, out of all the 28 serine amino acids that we find in chymotrypsin, this is the only serine that reacts with DIPF. And this is found inside the active site. And so that implies that it's this serine molecule, and the serine molecule has a number of 195. So if we count all the amino acids beginning with the first one along the entire polypeptide chain, this serine will be labeled as 195. So among the 28 serine residues found on chymotrypsin, only serine 195 actually reacted with this irreversible inhibitor, diisopropyl phosphofluoridate. And so this implied that serine 195 must be the one that plays a major catalytic role in the active site of that enzyme. And so that's how we know that serine is the one that is involved in actually catalyzing the cleavage of the peptide bonds at these specific bulky hydrophobic amino acids. Now, the next question is, what is the general description of the reaction mechanism that takes place inside the active sites? Well, basically, it's a two-step catalytic mechanism, and we'll discuss the steps involved in much more detail in the next several lectures. In this lecture, we're simply going to paint a general description, a general picture of what takes place inside the active site and how serine is actually involved in breaking and catalyzing those peptide bonds. So we see that experimental data suggests that chymotrypsin uses a two-step mechanism to actually hydrolyze peptide bonds. Now, the way that scientists actually studied the reaction mechanism of chymotrypsin is by using a special type of substrate molecule that once we react that substrate molecule with the active site, the product that is produced gives off a color. And so we can study the absorption of light when that colored product is produced, and that will give us information as to what happens in that reaction when we use chymotrypsin. So once again, to monitor the reaction progress, we generally use a substrate that changes color when it reacts with the active site. So we have this special substrate molecule that when reacts with the serine inside the active site, it produces a product that has a certain color to it. And we can essentially monitor the color change and the absorbance of light as we produce that particular product. So if we take a look at the following graph, the y-axis is the absorbance of light of that product produced when the substrate is catalyzed by the active side, and the x-axis is basically the reaction progress. So at the 0 point, at time 0, we basically take our mixture of that special substrate and we mix it in with our enzyme, and now our reaction begins. And notice what kind of curve we actually produce. So we see that initially we have a very steep slope, a very large slope, and then the slope begins to level off. And so here the rate of the reaction is low, but here the rate of the reaction is very fast. Now, what exactly does that tell us about the reaction mechanism of chymotrypsin? Well, what it tells us is we have this initial fast burst stage, and that describes the first step of the reaction. And the first step of reaction, the actual binding of the substrate onto that catalytic serine of the active site, is a very quick step. But as soon as that step takes place, we have a second step, and the second step is a slow step. And so what happens is we have the initial very quick step taking place, and then eventually when the steady state conditions are reached, when the intermediate concentration doesn't change too much, we see that the slope begins to decrease, and so we have this relatively slow process taking place. And that's essentially a result of the second step of the reaction being relatively slow. So graphing the data that we obtained from experimental results, this shows us that the fast initial burst of the color product followed by a slowing down, and that's basically a result of us reaching the steady state condition, and that second step being so much slower than that first step. So to see exactly what happens, let's take a look at the following diagram. So in this particular diagram, we have the active side of the chymotrypsin, and this is serine 195. So let's suppose we add our chromogenic substrate. Chromogenic simply means when that substrate reacts to produce a product, that product will change color. And we can monitor the color change. We can basically determine the absorbance of light as the reaction progresses. So we have this chromogenic substrate, we have the active side, and so what happens in the first step is we have an acylation step taking place. So an acyl group found on this chromogenic substrate, so this purple group basically attaches onto the oxygen. And so when that takes place, this blue section, the amide product, is basically released. And it's this amide product, so RNH2, where the other H atom basically came from this group here. This is the thing that produces that colored product. And so as this reaction takes place, the acylation reaction takes place very, very quickly, and that's why we have a fast initial burst stage. And as this is produced, this creates a color change, and we can monitor that color change on the following graph. Now, once this reaction takes place, this oxygen of the serine in the active side is acylated. So this entire group is attached onto the oxygen as shown. And this is the covalent bond that is temporarily formed. Remember, chymotrypsin uses covalent catalysis, and that means we form a temporary covalent bond. So this intermediate is known as the acyl enzyme intermediate. And in the next step, we have the water molecule that joins in the active side, and it basically hydrolyzes the cleavage of the peptide bond. So it ultimately breaks this bond here, and it forms a bond between the oxygen and this carbon. And so in the second step, we have the deacylation step. So this acyl group is removed from that oxygen. The H from the water molecule is given to that oxygen on the serine, and so ultimately we regenerate that enzyme, and then we also form that hydrolyzed final product in which we now have a peptide bond that has been broken. And so this is the very quick step that takes place that accounts for this relatively steep slope. But as we reach the steady state conditions, as the intermediate concentration basically does not change, what begins to happen is this begins to level off. And this describes the fact that the second step takes place much slower than that initial step. And so this is the two-step catalytic mechanism that basically generalizes what takes place inside chymotrypsin. So the first step is the fast acylation step, in which the acyl group is added onto the enzyme, and then the amide is released. And it's the amide that is used to basically determine what the color change is. We use it to basically determine how much light is absorbed. And then in the second step step is a much smaller step, and in this step, we basically break down the acyl enzyme intermediate by using water as a nucleophile, and then we regenerate that enzyme, and we form that hydrolyzed final product. Now, in the next several lectures, we're going to look at a much detailed discussion as to what actually happens inside the active side and what the reaction mechanism actually is.